uh, in the context of metabolic networks, enzyme expression levels can be treated as regarded as parameters, or in signal transduction networks, external stimuli can be parameters. And major theoretical challenge is that for many reactions inside cells, we don't have a precise knowledge of reaction rate functions. And this raises the fundamental question. So can we understand the behavior of these systems solely from network structure alone? And it turns out that many properties of chemical reaction networks are determined by their network structure. And along this direction, we have developed two methods. And my collaborator, Professor Mochizuki, presented a seminar here last November, and he presented structure sensitivity analysis. This is a method of determining the steady-state sensitivity based on network topology. And yeah, so the other one is structure bifurcation analysis. This is the main topic. But this structure bifurcation analysis was built on the structure sensitivity analysis, and some of you may have missed his talk. So today I'd like to begin with a review of structure sensitivity analysis, and then move to the bifurcation analysis. Okay. Okay, so to introduce some notation, I quickly review the dynamical system of a chemical reaction network. Yeah, the dynamics is described by ordinary differential equations shown here. Here, Xm is the concentration of chemical species M. And here on the right, Ri is the reaction rate function of reaction I, which depends on parameter Ki, and also depends on some concentrations, of course. And finally, this matrix S is the so-called stoichiometric matrix, and this is usually integer matrix, and yeah, which encodes the network structure. And regarding reaction rate functions, it is common to assume specific forms such as mass action kinetics or Michaelis-Menten kinetics written here. However, as I mentioned earlier, since we often lack precise knowledge of true functional forms inside of cells, so in our approach, we don't assume any specific functional form. Instead, we only assume that each reaction rate depends on its substrate concentrations. So yeah, so for example, in this toy network, I assume that R2 depends on the concentration of A, because A is the substrate of the reaction 2. Okay, so I assume only this such a structural information only. And now let's review the structure sensitivity analysis. And this is a method of determining steady-state sensitivity, namely how the steady-state concentrations depend on parameter, for example, Ki. And here I sketch the method. Okay, so we begin with the steady-state condition. This condition implies that the reaction rate vector R must be in the kernel space of matrix S, right? So we can expand the reaction rate vector R in terms of the kernel vectors of the basis vector of the kernel space S. So here C alpha C alphas are kernel vectors, the basis vectors of the kernel space, and mu alpha as the coefficients. And by differentiating this equation with respect to a parameter K, we obtain this expression which captures how the steady state is affected by a given perturbation. And this can be written in using rewritten compactly like this using a matrix A. And here the matrix A is defined by this equation. So A consists of two parts. The left part consists of reaction rate derivatives, and the right part consists of the kernel vectors of the stoichiometry matrix. And this expression implies that the sensitivity can be obtained by computing the inverse of this matrix A, right? With minus sign. And the crucial point is that the matrix A is typically a sparse matrix that reflects the underlying network structure. And A inverse is also a sparse matrix. And whether each sensitivity is zero or non-zero can be determined only from the structural information of the reaction system. So I'd like to illustrate this point in a simple example. Okay, so for this simple network, the matrix A is given as shown here. So derivatives and kernel vectors here, and the sensitivity is given by its inverse. Sorry, here RIMs represent reaction rate derivatives. And usually reaction rate functions are monotonically increasing functions of concentrations. So small r's are usually positive. So we can determine these elements, the signs of these elements as shown here, okay? And yeah, below I'm just showing this result on the network. So for example, this triangle, so for example, in this case, this means that decreasing the parameter K4 will decrease the concentrations of A and B and reaction rates along this loop. And interestingly, the downstream chemical C is not does not respond to the perturbation. Okay, so in this way, even though the reaction network itself is connected, only a limited part of the network is affected by a given perturbation. So the response, non-zero response is localized in a particular part of the network. And we have found a general theorem that determines which part of the network will exhibit non-zero responses to a given perturbation. And here's the theorem. Okay, so suppose that a subnetwork gamma satisfies the following two conditions. Sorry, so the gamma consists of a subset of chemicals, V, and a subset of reactions, E, okay? And the first condition is that the reactions inside gamma include all reactions whose rate functions depend on chemical species of gamma. So this means that, for example, let's suppose that this this node, this chemical species is inside in the gamma, then these two emanating reactions must depend on this concentration. So we need to include these two reactions as members of E gamma. Okay, this is the first condition. Secondly, we calculate a kind of Euler characteristic. So the index is defined like this. So it is given by the number of chemical species inside gamma and minus the number of reaction edges reactions inside gamma plus the dimension of kernel space restricted on gamma. And graphically, these three terms corresponds to nodes and edges and cycles inside gamma. Okay, and let's suppose that this index is equal to 0. And we call such a subnetwork a buffering structure. Because for such a subnetwork gamma, we can show that any parameters inside gamma do not affect the outside of gamma. Okay, so the concentrations outside of gamma is not affected by the parameters perturbation inside gamma. So such a subnetwork can confine the effect of the perturbation inside it. And here are examples. Okay, so yeah, in the second, so let's see the second case. So yeah, these two subnetworks closed by orange lines are buffering structures, but let's focus on, let me explain it in the second case. So yeah, you can easily confirm that the first condition is satisfied for this subnetwork. And about the second condition, the index, yeah, we have two chemical species AB and 3 reaction edges. So we have 2 minus 3 here. And there is a single loop, one loop here, cycle here, 2, 3, 4. So we have +1 here. So the index is 0. So this subnetwork is a buffering structure. Okay. And consistent with this theorem, the perturbation of reaction 4, I mean parameter K4, affects only A and B and the reaction rates inside this structure, and the concentration of C does not respond to this perturbation. Yeah, because C is the outside of the buffering structure. Okay. And I don't go into the proof, but basically these two conditions imply that matrix A as well as its inverse have a block triangular shape as shown here. And each square block, so this square block corresponds to the buffering structure, and this square block corresponds to the outside of the buffering structure. And 0 elements appear on the lower left corner of A inverse, and this means 0 sensitivity outside gamma. So this corresponds to the statement of the theorem. Okay. Okay. So yeah. So far I have reviewed the structure sensitivity analysis. So in the remaining time, I'd like to turn our attention to the main topic, bifurcation phenomena. Yeah. So as I mentioned earlier, we can compute the sensitivity by taking the inverse of matrix A. But the natural question is, what happens if the matrix A becomes singular at a certain point in the parameter space? And the answer is, as I will explain soon, the singular A indicates occurrence of a bifurcation phenomena. Okay. Yeah, and yeah, I think most of you are familiar with bifurcation analysis, but let me briefly explain basic facts. Okay, so yeah, as you know, a standard way to analyze the stability of a steady state is to compute the Jacobian around the steady state. And when steady state bifurcation occurs, the Jacobian becomes singular because one of the eigenvalues becomes 0. And in this example, we have x and y, and so Jacobian is a 2 by 2 matrix, and its determinant is proportional to square root of k. K is a parameter of this system. And so we can conclude that the steady state bifurcation occurs when K becomes 0. And these plots shows the steady state concentrations as a function of parameter K. Yeah. So yeah, as you can see, at K equals 0, uh, Y exhibits bifurcating behavior. Yes, and in this example, only Y exhibits bifurcation behavior, and X not. And generally, the bifurcating variables are determined from the support of the null vector of the Jacobian. And in this case, the null vector of the Jacobian at the bifurcation point is given by 0, 1. And yeah, and so from this, we can conclude that only Y exhibits a bifurcation. Okay. Okay, so let's apply these basic facts to a chemical reaction network. Okay, the Jacobian of a chemical reaction network is given by this. So it's a product of the stoichiometry matrix and the reaction rate derivatives. And you will soon notice that our matrix A is defined exactly from the same data, S and reaction rate derivatives. And in fact, these two matrices are essentially equivalent. And specifically, we can show that their determinants are proportional to each other. So a bifurcation point can be characterized by a singular A instead of singular J. Okay. So what is the advantage of using A instead of J? Yeah, the answer is that our matrix A reflects the network structure of a reaction network. Specifically, when a buffering structure exists in the network, as I briefly mentioned earlier, the matrix A has a block triangular form, as shown here. Here, gamma is a buffering structure, and gamma bar is the outside of the buffering structure. And this block triangular form implies that determinant of A is factorized into two factors. One is associated with the buffering structure, and the other is associated with the outside. Okay. And so this form implies that bifurcation occurs either when one of these two blocks yeah, when either one of these two factors becomes singular. Okay? So there are two cases. And the parameters that can induce a bifurcation differ between these two cases. The factor associated with the buffering structure depends on all of the parameters. So all of the parameters, so any parameter can potentially trigger the first type of the bifurcation. But the factor associated with the outside of the buffering structure depends only on the parameters outside of the buffering structure. So only parameters outside of the buffering structure can induce this type of bifurcation. Okay, and furthermore, the chemical species that exhibit bifurcating behavior also differ between these two cases. And when specifically when the buffering structure becomes singular, then only chemical species inside the buffering structure exhibit bifurcation. On the other hand, when the outside becomes singular, all chemical species exhibit bifurcation. Okay, so yeah, this difference arises because the support of the null vector of matrix A differ between these two cases. in the first case, the support is only on the buffering structure, but in the second case, the support is both on gamma and gamma bar. This can be easily confirmed from this block triangular shape. Okay. This is a structural consequence. So this should hold for any kinetics. Okay. So let me illustrate these results. So let's consider this network. And here arrows are reactions. So we have 5 reactions, and blue dotted lines indicate negative regulation. So for example, reaction 5 is negatively regulated by the concentration of B. Okay. And there is a single buffering structure in this network. So the determinant of A is factorized into 2 factors, the buffering structure and the outside. Okay. And in particular, and when gamma becomes singular, only chemical species inside gamma, namely only chemical A, exhibits bifurcation. On the other hand, when gamma bar becomes singular, all chemical species ABC exhibit bifurcation. This is a prediction of our method. And to demonstrate this prediction, here I assumed specific kinetics and numerically computed steady state concentrations as a function of a parameter in 2 cases. In the first case, when I change this parameter, then the determinant of the buffering structure becomes 0 at a certain point. And at this point, only A exhibits bifurcation, and B and C not. And in the second case, when I change the parameter value, determinant of gamma bar becomes 0 at 2 points. And at each point, all chemical species exhibit bifurcating behavior. Okay. So these results are consistent with the prediction from the structural argument. And these results can be extended to a network with multiple buffering structures. And I'd like to explain the general procedure of our method in this slide. So for a given network, we first enumerate all buffering structures. And then we construct a hierarchy or inclusion relation of these buffering structures. And then our method implies that the determinant is factorized into the multiple factors, and each factor corresponds to a particular buffering structure. And secondly, the property of buffering structures implies that each determinant factor depends on parameters of the upper subnetworks in the hierarchy. For example, A gamma 3 depends on parameters of gamma 3 and parameters of this top subnetwork. And lastly, when the bifurcation occurs, one of these factors becomes 0. And when a subnetwork becomes singular, bifurcation behavior is confined within its lower subnetworks. For example, if this blue subnetwork becomes singular, then chemical species inside blue and red subnetworks exhibit bifurcation. Okay. Okay. So here I demonstrate our method in a realistic network. This is a central carbon metabolism of E. coli, which consists of approximately 50 reactions. And there are 17 buffering structures inside this network. So the determinant is factorized into 17 factors. And then by examining these 17 factors one by one, we found that only the factor associated with this red subnetwork has a possibility that it becomes singular. And all the other factors are positive definite. So we can focus on the red subnetwork. And our method implies that when this red subnetwork becomes singular, only chemical species inside red and blue subnetworks exhibit bifurcation. And here I demonstrate this prediction by numerical simulation. Okay, so here each panel shows a bifurcation diagram of a particular chemical species. And as consistent with our method, bifurcation is observed only for chemicals in the red and blue subnetworks around here. Okay, so in this way, by using our method, we can analyze, we can determine qualitative aspects of bifurcation, even for large network. Okay. So let me summarize my talk. So today I explained the two roles of buffering structures. The first one is the localizing property of sensitivity, and the second one is the localizing property of bifurcating behavior. And these structural methods are useful for analyzing complicated networks, and also suggest that certain properties in biological networks may emerge solely from network topology. Okay, so finally, I'd like to thank all of my collaborators, and thank you very much for your attention. And I'm happy to take questions.